Thanks everyone for joining us for the September 2024 version of the seminar on physical genomics. My name is Benjamin Keene. I'm the Senior Program Administrator here at CPGE at Northwestern University. We're really excited to welcome our invited speaker today, Dr. Dave Thirumalai, who is the Colley Welsh Regents Chair at the University of Texas at Austin. Dr. Thirumalai's research interest lies in understanding the physics of chromosome folding, with a particular focus on the role motors play in affecting the structures of mitotic chromosomes. In addition, he's also developing theoretical and computational methods to describe collective cell migration, which have potential applications to the physics of cancer. Dr. Thirumalai, thank you for taking the time to share your work with us today. For our audience, we'll have a Q&A session at the end of today's talk, or if you want, you can just type questions into the Zoom chat anytime you think of them, and we'll address them at the end. If you'd like to learn more about the research and training programs here at CPGE, you can visit us at physicalgenomics.northwestern.edu. We have a video archive of all our monthly seminar talks and our other symposia on our YouTube channel, or you can follow us on Twitter, X, or Instagram. So at this point, Dr. Thirumalai, I think if you want to share your slides, we can turn the floor over to you. I think I need to stop sharing first, though.
Yeah.
Okay, there you go.
Oh. No, this is not. Sorry, I got it. I clicked the wrong guy. Share. I suddenly don't see my.
Sometimes if it shows all your apps, you have to scroll down.
Yeah.
There we go.
You see?
Yeah.
Okay. I wanted to go to slide up. Oh, I want. Okay, let me stop. Yeah, this is possibly the most important slide, and I should start with it. So I want to tell you about the role of motors in genome folding. It's a relatively new topic even in the experimental world, although I'll show you historically some aspects of it has been known for quite some time. This work was done by three excellent people. At least one of them, I think, is on the Zoom call. His thesis was partly on the subject. Guangxi, he was a student, and he left, and then he's come back as a postdoc. Atreya is now in UCSF, and Ryota was part of his thesis was on roller motors as well in genome folding. He is now a postdoc in Max Planck in Dresden. And it's based on a couple of papers that I'll discuss, three of them in particular. I should tell you that there are lots of other people before, after, and during our work. And in particular, from a theoretical perspective, John Marco was among the first to actually translate some of the ideas of loop extrusion, which is supposed to, in fact, be involved in the folding of the genome. And there are others as well, and I shall mention that. So this is a summary of my talk, if you wish. I'm going to show you a theoretical work that allows you to calculate the force velocity and loop size distribution of a single condensing motor when it acts on DNA without its nucleosomes, and because those are the experiments that are done in 2018 by Case Decker. And of course, in reality, there are multiple motors involved in organizing the genome, and we focus in particular on mitotic chromosomes and the role that multiple motors play in compacting the DNA as the cell cycle goes from interphase to the mitotic stage. And I'll tell you what are the kinds of structures that are generated in the mitotic chromosomes. And this, of course, takes motors into account implicitly, and I'll argue that these mitotic chromosomes can be thought of as structures with near zero curvature and no twist. All right. So most people here are non-molecular biologists, but but molecular biologists, when you tell them you're looking at a structure, they talk about all the heavy atoms. This is a ribosome structure, and they're determined by cryo-EM increasingly and X-rays. And one knows precisely what the static structure is, and the object of programs like AlphaFold algorithms are to predict these from sequence alone, and they have been, in fact, remarkably successful in the last several years. But that's not what is meant by structure, although it's used in the same way, I think. What is meant by structure in chromosomes is somewhat different, largely because chromosome as a polymer is gigantically large. It's the largest polymer probably that we know. It can vary in a number of monomers, which are just ATGC base pairs, from about 100 million to even close to a billion, depending upon the species and so on that you look at. So that means, A, you cannot know what the precise structure is, and B, from a polymer physics perspective, nor should you ask that question at all. And what you want to know is a coarse-grained description of what the structure actually looks like. And that's more or less what the experimentalist can do. And this began in earnest in 2009, although there were experiments like this done long before that. And these are the high-C experiments that Joop Dekker, who developed the technique when he was in Nancy Kleckner's lab in 2002, in fact reported starting in 2009. And now this has become essentially a household tool in getting at structures. So what did these experiments tell you? They tell you, and there's a bunch of chemistry involved. I don't want to get into this at all. If you know, you know. If not, it's not relevant. It tells you, after you look at, it's an ensemble average structure which reports on the probability that two loci that are separated by genomic distance are in spatial proximity. What is the probability that happens? And of course, there's some resolution involved in this. And the current resolutions are almost always stated to be much higher than they are. But we now know, let's say quite safely, that probably you can get resolutions on the order of 10,000 loci being lumped into one. That means even this is not the highest resolution, but you can actually get it not 10,000 base pairs, and how two such base pairs are at a distance, let's say on the order of the closest distance between them. Right? You can measure that, or you can infer that from these contact maps. So that's what they tell you. And the contact maps, roughly speaking, for the purposes that I'm going to use today, can be described in terms of competition between two major length scales. And one length scale is that it is known now for quite a while, in fact, 100 years maybe, that or more, that chromosomes can be roughly partitioned into active and inactive parts. One is the euchromatin, that's the active part that carries the genes, and that's what's expressed when the polymerase acts on it, along with transcription factors and all other things. And the other is heterochromatin, which is silent and plays an important role as well biologically. So the contact maps tells you, and these blues and reds are these chunks of chromosomes, and therefore you should view this interface chromosome as a copolymer, roughly speaking, consisting of two kinds, a euchromatin, a B chromatin, but they're not block copolymers, but they're copolymers with some regular sequence, and the sequences play, in fact, an important role. And there are two length scales. The length scale is on the genomic scale of the order of, say, few megabases. These chromosomes roughly undergo microphase separation into those which are rich in euchromatin and regions that are rich in heterochromatin. And then if you look at the diagonal, and these are called topologically associating domains, although there's no topology involved here at all. It's just, I don't know, word that came from somewhere and it's stuck. And along the diagonals, the various heterochromatin and euchromatin are even greatly separated or insulated from each other, and they're believed to play some role in transcription. So that's what they tell you, that these experiments tell you. And this ensemble average, but I mean, you have to look at about millions of cells and to get at these 2D maps. It's often plotted as a contact frequency, the probability of contact between two loci separated by some distance. And this turns out to be an experiment that I'll come back to quite a bit later in DT40 chicken chromosomes. So that's what we know from these experiments. The mitotic chromosomes, incidentally, the interface chromosomes are thought to be globular. So they're kind of like polymer globules. The distance between two loci separated by some distance scales is S. The distance between the loci to the power of 1/3. That's a compact globule. So mitotic chromosomes are helical, and these are the various renditions of the helicity that are reported in this very nice paper by Nancy Kleckner 3 or 4 years ago. And that these mitotic chromosomes are somehow helical was known over 100 some years ago, but now we know for sure it is, but we don't know what kind of helix it is, and that's still being actually debated. I'll come back to this towards the end. So the first, most of the talk that I'm going to tell you about is the loop extrusion. There are lots of aspects of loop extrusion. It's believed that this interface chromosome, from a transition from mitotic chromosome to interface, there are motors. And these are largely, but not exclusively, cohesin motors. And I'll show you what they look like in a second. They extrude some loop, and the loops, in fact, coalesce in some fashion to form this globular structure for interface and extended helical structure for the mitosis. Different kinds of motors are involved in the two structures, two phases of chromosome. The fact that the loops are extruded, and I'll show you a video, which is a cartoon, was first proposed by Naismith and in a review article about 24 years ago. But the first really sense into what that would mean in practice was proposed by Alipur and Marco in a paper that was published in 2012, and I'll briefly describe that in a second. That is followed by a couple of other groups. Erez Lieberman when he was leaving MIT at Harvard, something like that, and then Murney subsequently. So the basic physical picture that began with Marco's work, John Marco's work, and then was carried out further by these two groups, is that if you think of chromosome as a thread, then the cohesin motors, they're thinking about interface, falls on them randomly, and then they translocate along this chain, and the translocation stops when they encounter these architectural proteins, which are thought to be CTCF. So these are proteins tells you the stop signals, and then the loop is actually, loop extrusion comes to a stop. Now, of course, it can randomly go anywhere, and the loops can be of different lengths, as this one shows, and maybe this one shows as well. And so the idea is that the motor translocates, powered presumably by ATP and things like that, and that's the translocation model. And about 4 or 5 years ago, Marco worked out a little bit more the theory to produce some observables, and I'll mention that later. We actually have a different view, and this view is called the scrunching mechanism, and it's best illustrated by this animation, which you shouldn't believe really, because this is really a cartoon. And it also has the same picture. This single condensate, for example, lands on DNA, and loop is coming out on one side, while the motor itself is relatively stationary. Motor doesn't move; it just simply extrudes like something that threads it out of like a needle and a thread. Okay. That's called the scrunching mechanism, and I'll describe a little bit more about that. So in the first experiment, about 6 years ago, Kees Dekker in Holland did an experiment with tethers DNA between, which is on a cover slip, and it's pinned at two ends. There's a flow that's generated, which exerts a force, and of course ATP is involved in this. And then the loop is extruded, and this is the real video of that experiment. And you can see this is a loop, and you can look at this a little bit more carefully in that experiment. And in fact, there's no doubt that under this in vitro setup, the loops are in fact extruded by the condensation motor. It's not cohesin in that case, yeah. The assumption is that because of the architecture of condensation, there are possibly 2 ATP molecules that are consumed for every step or every time the loop is extruded, and then as the cartoon showed, the folding of the DNA, the extrusion of the loop, of DNA occurs by an asymmetric process. And what they could measure was, by doing this bunch of experiments, the rate of extrusion of this loop as a function of extension, which then translated into force. And what you have is a force velocity curve. It starts out at near zero forces at about 1,500 base pairs per second, and the motor stalls around 0.8, 0.9, say 1 piconewtons. So it's a very wimpy motor. It's a very weak motor. Stall force is quite a bit less than conventional motors that we know. So this movie this experiment, not the movie, caught my attention, and that's what Ryota and the other two sort of worked on. A little bit later, 4 years ago, they reported on the experiment using magnetic tweezers, and these setups are well known. I don't have time to tell you a lot about this. The DNA is actually tethered. You apply a magnetic field, you supply ATP. These are biotin streptavidin, and when you supply ATP, the distance between this magnet, the bead, which is stabilized by the magnet, and the surface is reduced, and the distance decreases. The distance decreases means at some moment the loop is extruded, and if you look at the trajectory, which is the position of this bead as a function of time, you see it keeps decreasing, and each time there's a decrease, and there's loop is extruded, the Z is less than 0 is extrusion, and you can see that the amount of loop that's extruded varies depending upon time and also the experiment. This is meant to show you that if there's no ATP, there's no loop extrusion. Okay, so what we wanted to do was, I'm going a little slow, so I'm going to skip this. I'll come back to this a little later. We had worked a lot on molecular motors, culminating in the reviews of modern physics that we wrote in 2020. And we were able to calculate for conventional motors like myosin, kinesin, and dynein, things like distribution of step lengths, which would correspond to distribution of loops that are extruded and the velocity and stuff like that, and compare with experiments. So we knew how to do these sorts of calculations. So we wanted to think about this in the context of a single condensate extruding a loop to understand quantitatively the kind of experiments that Case Decker reported. And so I'd known about this so-called scrunching mechanism. And if there's nothing else that you remember about this experiment, you should look at these two spectacular papers that appear in a completely different context, namely transcription initiation by bacterial polymerase. And there are two back-to-back papers in Science, and there's a single molecule FRET experiments. And what they showed was, in fact, the bubble opening, which is required before pole 2 can start synthesizing the protein occurs by the so-called scrunching mechanism. And I'll tell you a little bit more about this, but it's worth reading the abstract. These abstracts are almost identical. One of the authors is the same in both, and they are back-to-back papers in Science. They show the initial transcription procedure as scrunching mechanisms, in which polymerase remains fixed on the promoter, which is the DNA, and pulls down the DNA into its self-patched active center. So it's sitting there and it's crunching the DNA inside. And this is something that I knew quite a while ago, and in fact was the subject of a thesis of my former student. And we wrote a paper in collaboration with Seth Dawes, who was a Rockefeller. So what do they mean by crunch? How do they show this? So I do want to spend some time telling you about this. They did multiple FRET experiments by attaching fluorophores at various locations on both the polymerase as well as the DNA. And except for panels A and C, these are computer simulations that Jay Chen did, you can see the distances between the various label guys. This is on the polymerase, and this is upstream. This is on the polymerase, upstream. They don't change at all during the bubble opening. So they're stationary. However, if you label the polymerase as well as the DNA downstream of the center where the transcription starts, then the distance changes, it decreases, and then it plateaus out. Likewise, if you label a downstream DNA and an upstream location, that too, in fact, changes. So that means that the polymerase has to be stationary. It's only locations involving the DNA that, in fact, changes, and therefore that the polymerase actually scrunches this DNA into itself. So this is the mechanism that we want to employ. Okay, now back to condensation. Condensation is a pretty large motor. Cohesion, by the way, looks more or less like this. And for the purposes for this talk, I'll think of the motor head. There are two motor heads. They contain the ATP binding sites. These proteins that are attached here play a role in a manner that I don't fully understand, because the variations in that, in fact, produce variations in the functions of different condensate or cohesion motors. And then there's a hinge that's about 50 nanometers away. So you can think of this as 50 nanometers motor. And we know much less about this. For example, we don't know the full structures of the motor in complex with DNA, except some very rare cases, not in condensate, but cohesion a little bit. And we don't know much about the nucleotide states of these guys. So like all motors, the way in which you want to look at it, or when we looked at it, was there's a motor part that's just sitting stationary at the end once it falls on the DNA, but it binds ATP, undergoes ATP hydrolysis, releases the hydrolysis product, and the process extrudes loops, and that's what is summarized in these two ways. So this is a catalytic cycle. So in the beginning, there's no ATP to the bound states. ATP then binds, and we think this is the power stroke. in the process, it brings the distance between the head and the hinge close together. So R2, the distance between these two guys is less than R1. Then there's a hydrolysis of ATP. There's a product release, which is inorganic phosphate and ADP. And then eventually the ADP is released and you're back to the starting point and the cycle begins anew and you can extrude another loop. So that's what's happening in motor, where it's stationary, going through this allosteric transition that brings the head and the hinge close together. What's happening to the DNA trajectory? The DNA trajectory, the loop is extruded at each ATP step. There's some amount of Z that gets less than zero in the magnetic tweezer, and then there's loop extrusion. But occasionally, and just like other motors, it also coughs up the loop that's already extruded. There's a loop reversal that's not shown in this step, and it turns out that we can work out a theory for that as well, and we have done. It's in the process of being published. So those are words, and I don't want to show much math here, but the abstraction of this picture of the loop extrusion by scrunching mechanism goes like this. Let's say you have these 2 points, which are the head and the hinge, if you wish. The motor has fallen somewhere there, and the distance between these two guys is some value R1, and some amount the loop is already extruded. L0 can be 0 as well. And so there is a loop that would be extruded here because this is a semi-flexible chain. So if you pin these two ends, there is a length associated with this loop that you can calculate. And then because of this motor activity, ATP cycle in the process, R2 would become less than R1 upon ATP binding, and then there's a moment of the loop that's actually extruded. So what we want to know is how much loop is extruded when this distance goes from R2 to R1. And so once you know that, that's just a polymer part. That doesn't involve for you to know what's happening with the ATP. And one can compute that. And when you compute that, you don't need to know this at all. In fact, we didn't even bother publishing this paper. It's in the archive, but we use this, we and others use this for lots of other things. The loop, so there's some amount of length that's extruded per ATP consumed, and you can calculate this quite precisely. So that's a part that takes care of the architecture of the DNA, extruding the length of loop. But I need to have some bookkeeping about what the motor is doing. The motor is out of equilibrium, and the motor drives the system out of equilibrium by consuming ATP. And so I need to worry about the thermodynamics associated with the motor activity. And so in the process of working on the DNA, it does some work to either extrude the loop or stretch the loop or contract it. And that work is W0. And that happens in the absence of any external force. In the experiments, Case-Decker, either through magnetic tweezers or by flow, applies external force. And so if there is an external force, the motor will have to expend some energy to overcome that external load as well. So the currency that I have is hydrolysis rate. So N is the number of ATPs that's hydrolyzed. Mu is the hydrolysis free energy per ATP. And energy conservation tells you that these two have to be this equal. So W plus Q. So this is one criterion. Energy conservation relates the total currency that I have, how much I've actually spent, and there's always some dissipation involved, and which will eventually determine the efficiency of the motor. Once I know the thermodynamics, when I know the ATP cycle, I can use fluctuation theorem, and in fact, you can guess the formula to figure out what is the rate of extrusion of the loop, and that's given here, divided by some backward rate. And that's simply given by this formula, and therefore the rate of loop extrusion, you can guess this formula with dimensional analysis. It must be related to hydrolysis rate times some length scale time force. right? So that's what you would guess. And so we need to figure out what this length scale is, what the meaning is, what the value is. And then you would have an analytic expression for the velocity of loop extrusion as a function of an external force. All right, I'll come back to those length scale in a second. This is at the moment is a two-parameter theory. What do I not know? I do not know the hydrolysis rate. And I also don't know this length scale that's involved in extruding the loop, and I'll have to figure this out. So initially we use this as two parameters and fit the measured velocity of extrusion as a function of the relative extension, which is same as force for a semi-flexible chain. And that too goes back to the result due to John Marco and Eric Sidja some time ago. And when you do this fit, you find that we find that K0 is of the minimum value is of the order of 10 per second. The experimentalists tell us that the hydrolysis rate is 2 per second, so we're off by a factor of 5. But these experiments have never been repeated. They were done a long time ago. I don't really know. But we're within factor 5. The data, I must say, can be better. And Case is not doing this. Maybe somebody else will do this. But with these two parameters, we can also compute the distribution of the length of the DNA that's extruded each time we do an experiment. Each time we do an experiment, you'll get a different value, and that's what is being plotted here at the value of force, which is about half of the stall force, and you can see that the agreement with the experiments and an older experiment that Tatsuya Hirano, who did most of the works in the early stages on condensation 1 and 2, and the agreement is excellent, and everything is pretty good. So theory is fairly good, given that we didn't use much information. But we were bothered by the fact that we didn't know how to calculate the length scale. We still don't know how to calculate it, but we thought we could do a simple toy model simulation, and that's what we did. We pictured these connectors between the head and the hinge as semi-flexible chains with a flexible linker here. And we assumed that when ATP binds, the angle between the two heads, in fact, would decrease, and that triggers a stress which would propagate, and that propagation results in the closed transition. This is an occluded transition. When it comes here, it's a closed transition, and that's the allosteric transition that we are after. And we could then calculate for every single trajectory what the change in the length between the hinge and the head is. And then we do this simulation multiple times as a trivial simulation. You would get a distribution of these lengths from which we can actually calculate the mean length that goes into the theory. Okay, how does this work? I have to move a little faster. The bottom left is the experimental determination, or no, the right is experimental determination from Case Decker's group. This is published about 4 years ago. And the bottom left is a distribution that was obtained in the simulations. And when I first saw this, one of them, I'm not going to name who did it, showed me this. I thought he cheated. Therefore, I asked the other guy, I'm also not going to name that guy, to do this independently so that I could be sure. I didn't tell that guy. And it turns out that the value that we get, I mean, you should not take this agreement too seriously. The fact that it's of the order of 20 nanometers is sufficient. And therefore, now we can simulate this length scale, and therefore what used to be a two-parameter theory can be used as a single parameter theory. And this, of course, these lengths can actually be measured. So perfectly happy with these measurements, and let these calculations, And let me move. So it's worth for us anyway to think about how does this motor compare with the conventional motor. It turns out that the condensing motor is a weak motor. It's most likely close to the myosin 5. The velocity with which the stepping velocity in myosin 5 is 400 nanometers per second. And that's the extrusion number that you can get from the experiments for condensing. The stall force for myosin is about 2 piconewtons, and it's 1 here. Hydrolysis rate is about 300, 350 times larger in myosin than here. I don't know the origin of why this is so. There must be a structural reason at the end of the day. Okay. So it's worth thinking about these motors, not just in isolation, but as a theorist, you want to get a unified perspective on the functions of various motors. All right, now I think I was told I have till about 10 minutes at the most, and I want to tell you at the most actually. So I want to tell you about mitotic chromosomes, and let me go fast in this. Oops. So mitotic chromosomes, we want to extend this theory for single motors, so multiple motors. And of course, in cells, the motors are not tethered at the ends. The motors are where they are, and there are many of them. And so how, in fact, does this come about? So there was an experiment in 2018, now they're from Joop Dekker, who looked at the cell cycle of this DT40 using these high-C experiments, starting from the interphase, the G2 phase, till the mitotic chromosomes. It takes about 60 minutes. And you can see at the very end, within about 7.5 minutes, there are no signatures here, and there seems to be no structure like in the interphase where you have the possibility of the 2 length scales that I talked about before. So we wanted to think about how to calculate those contact maps from the theory of motors that I sort of briefly described. And if you look at the literature, there are 2 kinds of condensins, at least, condensin 1 and condensin 2. Cartoon-wise, they look exactly the same. We assume that the extrusion velocity in the absence of force is about 1,500 base pairs per second. They pause every once in a while, and the pause time is about 7 seconds. We got all of these data from HeLa cells, and we're going to apply this to DT40, which is a chicken cell. So who knows? The number of condensins in condensin 1 is about 2,500, and there are about 300 condensin 2s. The residence time of condensin 2 is much larger, means by a factor of 2.5. And condensin, there are more condensin 1s than condensin 2. The residence times are still much shorter than the time in which the mitotic chromosomes actually form. Therefore, this condensin 1 and they can come off and on quite rapidly. Rapidly means multiple times, right? Okay. So these are the postulated models. This is coming from the Dekker paper, courtesy of Murney, that they view these mitotic chromosomes as helical, where the central helix is made up of these motors that are stapled together, and these loops are emanating from the central axis. That's the picture. They use it to fit their high-C contact map. The different picture due to Nancy Kleckner, based upon 3D fluorescence imaging, which is live, but everything is fixed, it's an average, is that you should view these mitotic chromosomes as helices, because that was known, as I said, 125 years ago, I think. But the handedness of the helix changes almost periodically. So it's a left-handed helix followed by right-handed helix followed by left-handed helix. So that's the picture. I'm going to have to skip this. This is the data. The data is from Job Decker in this 18th paper. These are beautiful experiments, in fact. They did experiments with both the condensins present, only condensin 1, only condensin 2, and you can see the condensin 2 has some bumps that condensin 1 doesn't have, and this is in fact attributed to the presence of helicity, and the bump in fact moves quite a bit. Okay, so this is the extension to the motors, um, and you can see that we have multiple motors, and the motors are extruding loops, each of them quite independently until they encounter one another. Then it pauses for 7 seconds and continues. And the DNA here is modeled as a Rouse chain. And therefore, with this stochastic simulation combined with the polymer model, one could actually calculate these contact maps. Because I'm running out of time, I won't tell you the details about this. And if you calculate this, this is the dotted lines is a theory / simulation, and the rest are experiments, and the agreement is pretty good, but it's not all great, as I'll show you now. When you try to compare this for just condensin 1, the curves, they're pretty good. When you look at the data, compare the calculated data versus condensin 2, the very bumps that in fact gives rise to this pronounced helicity is absent, and therefore our theory, multiple mode theory, doesn't satisfy that. So I'm gonna skip. We have a method to calculate 3D structures, and from there, from Hi-C map, by 3D structures I mean coordinates of individual loci from which one can calculate what we want, and that's a result that's shown here for just condensin 1. This is just for condensin 2. This is for the wild type containing both, and this is for the HeLa cell at different experiment. If you look at the dashed lines, which are which are the experiments, and the dotted lines are maybe the other way around. Experiment, the agreement is extremely good. So there's no parameter in this theory at all, and we're able to completely reproduce that. So what do these structures actually look like? And please give me a couple of minutes because the last two things are important. So I've already mentioned this. We know it's helical, and therefore we can calculate an angular correlation function as a function of the genomic distance. And you can see the periodicity. And you can see when you have just condensin 2, the period is longer. The condensin 1, the period is shorter. So there are helical signatures even with condensin 1, but it's not as pronounced as condensin 2. Okay. One can look at the local helicity because that's what we were interested in. When you measure the local helicity, whether it's right-handed or left-handed, and you average all the conformations, you see that it is almost close to 0. And if you look at a single conformation, it's also slightly favors the left-handed, but the average helicity is quite small, in fact. So if you look at this stuff, it does look like it changes randomness, but not regularly, but the helicity changes randomly as you go across the chromosome. And this stuff was known long time ago. This notion of perversion, this is a term that Maxwell, in fact, introduced in his book, and he borrowed that from a mathematician called Listing. It just means that in this context, there's a reversal in chirality, and that's called perversion. And people who are older than 32 maybe know that these sorts of things in telephone cords a long time ago. So the prediction that we make is that the helix central axis is stapled by motors. The axis position changes quite a bit. As a result, the The loop positions changes, but the trace essentially this random helix where as you traverse along the axis, the helicity in fact changes randomly from left to right. So we call them random helix perversion. So it turns out that Darwin knew about this after Maxwell, and he wrote a book based on a lecture. I have it somewhere in the office where I'm giving this talk, and this book is got zero pictures, it's got zero equations, but it talks about this perversion, and it's called the tendril perversion. The plant biologists know this, and in fact, maybe many of you know this as well. And he talks about it in the context of this plant called Bironi, and he writes, I have more than once gone on purpose during a gale to watch a Bironi growing in an exposed hedge with its tendrils attached to the surrounding bushes, and as the thick and thin branches were tossed to and fro by the wind, the tendrils, had they not been excessively elastic, would have instantly torn off and the plant thrown prostrate. But as it was, the Bironi safely rode out the gale like a ship with two anchors down and with a long range of cable ahead of to serve as a spring as she surges to the storm. First of all, nobody writes like this anymore. Our papers are not read just because they're not as exciting as this poetic way of describing what he saw. And he sort of understood the importance of this tendril perversion in the context of plants. And I think we think at least Nancy's group and us that this is involved in the structure of mitotic chromosome. The question that is completely perplexing to me is from symmetry considerations, the structure that Nancy Kleckner proposed, that's a regular helix, is beautiful compared to this random helix perversion. Should nature care about this? And why would nature, in fact, choose a perversion, helix perversion, as a structure for mitotic chromosomes into something that's regular, as for example, Case Decker and Murney suggested? And the answer is really not clear. In fact, it's not even clear that this RHP or random helix perversion or the symmetric perversion is the right structure of mitotic chromosomes. This will require single cell data and imaging. It's coming, but it's not there yet. I think that's basically all I want to say to you. Thank you very much.